Kathleen Folbigg has always insisted she never killed her 4 young children.
Did you kill Caleb?
No.
Did you kill Catherine?
No.
Did you kill Sarah?
No.
Did you kill Laura?
No.
That's a position she maintained while exhausting all of her legal appeals and during 2 subsequent judicial inquiries. 20 years into a 25-year prison sentence, the justice system has now accepted there is reasonable doubt about her guilt.
I think the nation will look at the case of Kathleen Folbigg as a major miscarriage of justice.
Ms Folbigg was convicted of smothering her babies Caleb, Patrick, Sarah and Laura. The 4 died on separate occasions between 1989 and 1999. But at the time of her conviction in 2003, there was no way of knowing if gene mutations played a role in any of the deaths.
In fact, it was only in 2003 that the first draft of the human genome sequence was completed. Now, since then, it has become possible to sequence whole human genomes and exomes in a much faster and affordable way.
That's Professor Carola Vinuesa, who analysed sequences from DNA extracted from old heel prick samples of 2 of the Folbigg children.
She was concerned that the analysis of that genetic information had not been fully heard during the first inquiry into the convictions of Kathleen Folbigg. And it was those concerns and her resolute understanding of her science and her scientific integrity that in some ways didn't let her sleep at night.
Professor Vinuesa took those concerns to the Australian Academy of Science, where she is a fellow.
The more we looked at it, the more we too became concerned around the way science had not been heard in that first inquiry.
The science was complex. Research undertaken by Professor Vinuesa and complemented by independent experts around the world showed that Ms Folbigg and her 2 daughters possessed a rare gene mutation known as CALM2G114R,
which encodes for calmodulin. Calmodulin mutations typically are associated with cardiac arrhythmias that can cause sudden unexpected death in children and adults, both while awake and while asleep.
The second inquiry, which was instigated at the urging of prominent scientists, including 3 Nobel laureates, also heard medical evidence about epilepsy and respiratory problems, which could help explain the deaths of Ms Folbigg's 2 sons, Patrick and Caleb.
There is a reasonable doubt as to Ms Folbigg's guilt.
Retired Chief Justice Tom Bathurst KC accepted that the weight of the evidence cast reasonable doubt about Ms Folbigg's guilt, recommending her pardon. A key difference in the way the second inquiry considered medical evidence was the role played by the Australian Academy of Science.
I think now that with this new evidence, it's beyond reasonable doubt.
This is a clear indication that the independent scientific advice has an important role to play in the justice system.
The Australian Academy of Science was granted leave by the inquiry to act as an independent scientific advisor to it. I think unique to this case, and the role the Academy's been able to facilitate, is that the inquiry's been able to hear from a range of experts from the right subdisciplines to give a complete picture of the sort of science that is complex, and some of it's still emerging, that's been able to shed light on the possible causes of death of the 4 Folbigg children.
The Academy believes the case serves as a dramatic example of the need for a more formal mechanism to re-examine cases after appeals have been exhausted when new science is revealed.
Science is always emerging. Nothing is going to stop the advancement of knowledge, nor would we want to do that. It would be an enormous missed opportunity if we couldn't bring about some of the law reform that's needed so that science can more effectively inform the justice system.